Hello again. In this video we will continue to learn about genetic inheritance. In genetic inheritance, not all inheritance follows Mendelian inheritance. For example, codominant alleles, incomplete dominant alleles, multiple alleles, polygenes, sex-linked genes, and linked genes. Firstly, let's look at codominant alleles. Based on Mendel, in heterozygous condition, dominant allele will express its phenotype, meanwhile recessive allele will be hidden. But, in codominant alleles, both alleles are dominant and are fully and equally expressed in heterozygote. For example, the inheritance of human MN blood groups. There are three types of blood groups which are M, N, and Mn. Alleles that control Mn blood groups are Lm and Ln. For blood group M, the genotype is homozygous LM-LM. For blood group N, the genotype is homozygous LN-LN. For blood group Mn, the genotype is heterozygous LM-LN. Blood group Mn is an example for codominant alleles. In heterozygous condition, both Lm and Ln allele are dominant. Therefore, both alleles expressed their phenotype fully. Example of question can be asked is: a male with M blood group and a female with N blood group got married. By using a genetic diagram, show genotype and phenotype blood group of their children, phenotypic ratio and genotypic ratio of their grandchildren, if their son marries a female with Mn blood group. To answer this question, you have to draw a genetic diagram. For parents, M blood group LM-LM is crossed with N blood group LN-LN. Each parent produce gamete. Fertilization between gametes will produce F1, where all F1 are heterozygous LM-LN with Mn blood group. So the answer for the first question is: the genotype is LM-LN. The phenotype is Mn blood group. To answer the second question, heterozygous son LM-LN is crossed with heterozygous female LM-LN. Each parent produce gametes. Fertilization between gametes will produce F2. F2 produced are LM-LM with M blood group, LM-LN with Mn blood group, LM-LN with Mn blood group, and LN-LN with N blood group. From the genetic diagram, the genotypic ratio is 1 LM-LM, 2 LM-LN, to 1 LN-LN. Meanwhile, the phenotypic ratio is 1 M blood group, 2 Mn blood group, 1 N blood group. Second inheritance that is not following Mendelian inheritance is incomplete dominant alleles. Based on Mendel, in heterozygous, dominant allele will express its phenotype, meanwhile recessive allele will be hidden. But, for incomplete dominant alleles, neither of the two different alleles is fully dominant, therefore in heterozygous, an intermediate phenotype will be expressed. For example, flower color in Antirrhinum species, or snapdragon. The characteristic is controlled by two alleles, CW and CR. For red flower color, the genotype is homozygous CR-CR. For white flower color, the genotype is homozygous CW-CW. In heterozygous CR-CW, the flower color is pink. This is an example of incomplete dominant alleles, where both alleles in heterozygous is not fully dominant over each other. Therefore, an intermediate phenotype will be expressed. Pink is an intermediate phenotype, a blend of red and white. Example of question can be asked is: construct a genetic diagram of crossing between heterozygous F1, to show incomplete dominant alleles, including the phenotypic ratio, 1, 2, 2, 2, 1. And genotypic ratio by using Antirrhinum species, a snapdragon flower color. To answer this question, you must construct genetic diagram start from parent to show the heterozygous F1. P is red flower CR-CR crossed with white flower CW-CW. Each parent produce gamete. Fertilization between gametes will produce F1, where All F1 are heterozygous CRCW with pink color. Then, self-cross the F1 to show the F2. F2 produced are homozygous CRCR with red flower, heterozygous CRCW with pink color, heterozygous CRCW with pink color, and homozygous CWCW with white color. So, from the genetic diagram, the genotypic ratio is 1 CRCR, 2 CRCW, 1 CWCW. Meanwhile, the phenotypic ratio is 1 red, 2 pink, 1 white. Another inheritance that doesn't follow Mendel is multiple alleles. Based on Mendel, 2 alleles, dominant and recessive, control a characteristic. But, in multiple alleles, 3 or more alleles control a characteristic, but only 2 alleles will occupy a gene and determine the phenotype. For example, human AB-O blood group. There are 4 blood groups, A, B, AB and O. This characteristic is controlled by 3 alleles. IA and IB, dominant allele, and small letter I, recessive allele. For blood group A, the possible genotype are homozygous dominant IAIA, or heterozygous IAI. For blood group B, the possible genotype are homozygous dominant IBIB, or heterozygous IBI. For blood group AB, the genotype is heterozygous IAIB. And, for blood group O, the genotype is homozygous recessive II. For blood group A and B, homozygous dominant and heterozygous express the dominant phenotype. Blood group AB is another example of a codominant allele. Because both alleles are dominant, therefore both alleles express its phenotype. For blood group O, homozygous recessive II, express the recessive phenotype. Example of question can be asked is: construct a genetic diagram to show the possibilities of the offspring's blood groups, if a heterozygous A blood group father married to a heterozygous B blood group mother. To answer this question, you need to construct a genetic diagram. Parent of heterozygous A blood group is crossed with heterozygous B blood group, producing 4 types of F1, which are AB blood group, A blood group, B blood group, and O blood group. The F1 produced shows the possibilities of their offspring in every pregnancy. Therefore, when both parent are heterozygous for A and B blood group married, there are 25% possibilities that their children will be A, B, AB, or O blood group respectively. Next inheritance that doesn't follow Mendel is polygenes. Based on Mendel, 1 gene controls 1 characteristic. But, in polygenes, there are 2 or more genes control 1 characteristic. For example, like human skin color and height. Let's look at this diagram which use human skin color as example. There are 3 genes control human skin color. Crossing between heterozygous will produce wide range of skin color. Less individual with homozygous recessive genes which express fairest skin color. And, less individual with homozygous dominant genes express darkest skin color. Meanwhile, more individual with heterozygous genes express intermediate skin color. This is due to cumulative effect of polygenes, where changes in one gene will affect the phenotype, therefore, wide range of phenotypes are produced. Next is sex-linked genes. Based on Mendel's law of independent assortment, alleles are segregate independently into a gamete during meiosis. But, in sex-linked genes, allele or gene is linked with sex chromosomes and passed together into a gamete during meiosis. So, sex-linked genes is a gene that located on sex chromosome. There are 2 types of sex chromosome, X chromosome, and Y chromosome. X chromosome size is larger than Y chromosome. Therefore, mostly allele or gene is linked to X chromosome. Female has XX sex chromosome. Meanwhile, male has XY sex chromosome. Example used is hemophilia. Hemophilia is an inherited condition that affects the blood's ability to clot. Hemophilia allele is recessive, small letter h. Effect of hemophilia allele to female are: homozygous dominant XHXH is normal female. Heterozygous XHXH is carrier female. And homozygous recessive XHXH is hemophiliac female. Meanwhile, effect of hemophilia allele to male are either normal or hemophiliac. Unlike woman, no carrier for male. If X chromosome is linked to dominant allele and not recessive allele, the male is normal. But, if the X chromosome is linked to hemophilia recessive allele, a hemophiliac male will be formed. Male has higher chances to suffer the effect of X-linked genetic disease than female, because male has one copy of X chromosome only. Example of question can be asked is: a normal man married a hemophilia carrier woman. By using a genetic diagram, show the probability of their children for every time she get pregnant. The probability of their second child to be hemophiliac. The probability of their daughter to be a carrier. To answer this question, you need to construct a genetic diagram. Crossing between normal man and carrier woman will produce normal daughter, carrier daughter, normal son, and hemophiliac son. Based on the genetic diagram, the answer for the first question is: probability of having normal daughter is 1/4 or 0.25, carrier daughter is 1/4 or 0.25, normal son is 1/4 or 0.25 and hemophiliac son is 1/4 or 0.25 for every pregnancy. The answer for the second question is: the probability of their second child to be hemophiliac is 1/4 or 0.25. For this question, doesn't matter how many children they have, the probability of having hemophiliac child is always 1/4 or 0.25. And, the answer for the third question is: the probability of their daughter to be a carrier is 1/2 or 0.5. For every pregnancy, probability of each daughter to be normal is 1/2 or 0.5 and 1/2 or 0.5 to be a carrier. Only among daughter are considered, not among the children. The last inheritance in our syllabus that doesn't follow Mendelian inheritance is linked genes. Based on Mendel's law of independent assortment, alleles are segregate independently into a gamete during meiosis. But, in linked genes, genes are linked in the same chromosomes and passed together into a gamete. This diagram shows not linked genes, because the genes are located on the different chromosome. While, this diagram shows linked genes because genes are located on the same chromosome. The method to write a genotype for linked genes is different from a no-linked genes genotype. Genotype for linked genes is written using slanting method, or Morgan's method. But usually, slanting method is mostly used. Due to the two genes located within same chromosome, effect of crossing over may be occurred or not in the gametes. If there is no crossing over occur in a linked genes, gametes produced are all parental gamete. Meanwhile, if there is crossing over occur in a linked genes, parental gamete and recombinant gamete are produced. Parental gamete is a gamete that has a genotype identical to parental chromosome. Recombinant gamete is a gamete with recombinant chromosome due to crossing over. In a test cross of linked genes without crossing over, heterozygous parent will produce parental gametes only. Fertilization between gametes will produce parental offspring only. Crossing of linked genes without crossing over produce no variation, where all offspring are identical to parents. In a test cross of linked genes with crossing over, heterozygous parent will produce parental gametes and recombinant gametes. Fertilization between gametes will produce parental offspring and recombinant offspring. Crossing of linked genes with crossing over will produce less variation, because parental offspring are always produced more than recombinant offspring. This is due to the linked genes that located at the same chromosome and have tendency to be inherited together. Crossing over may occur but less frequent if compared to not linked genes. Parental offspring is offspring that has genotype and phenotype identical to parent. Recombinant offspring is offspring that has genotype and phenotype not identical to parent, due to crossing over. Example that is used in linked genes is body color and wing size in Drosophila melanogaster or fruit flies. Crossing between true breeding gray body and normal wing, with true breeding black body and vestigial wing, will produce F1 offspring where all are heterozygous gray body and normal wing. Test cross of F1 will produce F2 which are heterozygous gray body and normal wing, a parental phenotype. Homozygous recessive black body and vestigial wing, a parental phenotype. Heterozygous gray body and vestigial wing, a recombinant phenotype, and heterozygous black body and normal wing, a recombinant phenotype. Offspring with parental phenotype also known as parental offspring, and offspring with recombinant phenotype is known as recombinant offspring. Notice that, from the genetic diagram, the genotypes of linked genes are shown with slanting method. From the test cross, observed number of offspring shows that parental offspring has larger number compared to recombinant offspring. Based on the observed number of offspring too, the test cross of the F1 does not show ratio of Mendel's dihybrid test cross of 1 to 1 to 1 to 1. This is another reason why linked genes disobey Mendelian inheritance. As a conclusion for linked genes with crossing over produce less variation. Because parental offspring are always produced in large number compared to recombinant offspring. This is due to genes that are linked in same chromosome and has high tendency to be passed together into a gamete. The last subtopic in Chapter 4 is genetic mapping. Genetic mapping is the process of establishing the locations of genes on the chromosomes. It is only applied when the genes are linked in the same chromosomes, and crossing over had occurred. Please take note that it is only applied for linked genes with crossing over. This is an example for genetic mapping on a chromosome. Gene A, B, C, and D are located in the same chromosome. By using genetic mapping, we can determine the position of each gene. If the genes are situated further apart from each other, the possibility of crossing over to occur is high. Crossing over during prophase 1 of meiosis will produce recombinant gamete. If the linked genes are situated very close to each other, it cannot be separated by crossing over during meiosis. Therefore, even though crossing over occurred during prophase 1 of meiosis, combination of gametes remains same as the parent. The crossing over frequency is directly proportional to the distance between the two genes. The higher the crossing over frequency, the further the distance between the two genes. We can calculate crossover value or COV by using a formula. Unit used for COV is %, where 1% COV is equivalent to 1 map unit, while unit used for distance between genes are map unit or centimorgan. Example of question can be asked is: below are the offsprings from a test cross done on Drosophila species with gray body and normal wings. Calculate the distance between gene control for color body and wing size. Assume allele for body color is B or B and allele for wing shape is V or V. To calculate the distance between genes, you must calculate the COV first, by using the COV formula. From the question we can determine which are parental offspring and recombinant offspring, based on the offspring number. Larger number are parental offspring, while smaller number are recombinant offspring. By knowing this, we can calculate the COV, which are 17%. To state the distance between the genes, you need to convert the COV value unit into unit used for distance. So, the distance between gene control for color body and wing size is 17 map units or centimorgan. Second example is P, Q, R, and S genes are linked. COV for pairs of genes are as follows. Map the chromosome. Assume this is a chromosome. Tips to answer is, start with the highest COV. R and Q has the highest COV, followed by P and Q. Then, R and S. Then, Q and S. And lastly, is P and R. Now, all genes are mapped on the same chromosomes based on the distance between them. Well class. That's all about Chapter 4 Genetic Inheritance.